Hello everybody, it's time to start our new lecture from biochemistry and biophysics. I think we should wait a little bit because more and more people want to join us. Okay, I will start because for sure you will join the lecture for the next 1 hour and a half, so what to do. Okay, so today I want to continue the subject connected with biomolecules. Last time I was talking about lipids and sugars, carbohydrates, and I will continue this subject today. But first, I want to present you some topics for revision because one of the students from your group asked me to present some topics because this person want to start learning, and it was a good idea. So I can show you some topics which you can use for your during your learning, of course. Okay. So first, I should describe you how you should prepare to the final test. Final test means not that it will be just a test. Of course, I will prepare a few questions of single choice, so you will.
Excuse me, ma'am.
Sorry?
The sound system is really bad. It's echoing.
What system?
Your sound system.
Really?
Yes, we can't even listen to it.
I can.
But well, maybe I will put the voice of my speaker. It will be louder. Okay. Now it's better.
It's still quite not better, but much better from the first time.
Okay.
I think it's a problem because some of us are okay.
Okay.
Maybe it's because this is a really huge group of students. I don't know. I hope the rest of the students can hear me. Okay, so now please switch off your speakers. Maybe they disturb as well. In my in my talk. Well, so I will start once again. Where is my, okay, where I should find my presentation once again. What's going on? Okay. Okay, so it's really hard to have a lecture with so many students, but I hope you will be able to at least to see my presentation and of course to hear me. So here on this slide, you can find some topics for revision. So as I started to describe, the final test will have a few questions of single choice, but just only few, and the rest of questions you will need to write something from yourself. So this is a kind of open question, and in this case you should write some definition or compare some properties or list some kinds of bonds or mechanisms, things like this. That is why I prepared for you such topics, so I can briefly describe them. In first, you should write some definitions of biochemistry and biophysics. It doesn't mean that you should prepare this definition which I showed you, but you can use your own words, because the most important is if you understand the meaning of these terms. Then the next one, molecular organization of the cell. So you remember I was talking about some different levels of organization in our body. So I started with atoms and then molecules, then tissues, organs, systems, and so on. So this you should know. Then we have information about different kinds of cells. So the most important is comparison. So you can compare prokaryotic and eukaryotic cells as well as plant and animal cells. To learn this, you can prepare some tables, or just you can write down the most important information in your notebooks, and it will be good for learning. Then we have main types of transport across cell membranes. Here, just you should remember all these kinds of transport and just the main idea of each transport. So 1-2 sentences, it will be enough. Then definitions of electrocardiography and electrocardiogram. I was talking about this as well. And then chemical bonds. So for sure, you will find such question. Please list all kinds of types of chemical bonds, and you should be able to describe each bond, but also just the main idea. So what is the mechanism of formation of such bonds? Then I was talking about pH. So you should know the meaning of this parameter, the scale in which we can measure pH, and the examples of indicators. I was talking about this as well. Then I was talking about anabolic and catabolic processes. So you should be able to compare these both processes. So what are the similarities and differences between them? Then now we are talking about different structures of main kinds of biomolecules. So this I will explain you later when we will finish this this part of our lecture, what you should learn, should remember, but for sure such meanings like saturated and unsaturated fatty acids, what are the triglycerides, what is the structure. Then today I will talk about mono, oligo, and polysaccharides. So you should be able to find the difference between them. And then I will talk about proteins, amino acids, then during the next lecture about the structure of nucleic acids, like DNA, for example. So such information, and you can find some names to explain names of different chemical compounds from different groups of biomolecules, like lycopene or table sugar, also cellulose, starch, glycogen, inulin, collagen, things like this. And then during the next lecture, I will talk about nucleic acids, so also the structure and the main functions of such molecules. I know that you have a lot of time because this is just the middle of our lecture, but maybe it will be useful for you to prepare some revision of such topics. And as we have only 30 hours of this lecture, I show you only the most necessary information from biochemistry, chemistry, biophysics. Of course, sometimes this may be not too much, and you are interested in more information. So I recommended you at the first lecture some books. You can download then these books from internet because they are free, so everybody can download them. But of course, such books, they have hundreds or thousands of pages, so this is not possible to learn or even to read everything. But you can find some special topics, especially interested for you. And also, when you will learn, you can find plenty of different films in YouTube. And here on the next slide, you can find some links to such films from 2 subjects. 3, they describe the types of cells. So they are really interesting, not very long. You can watch them. And the 3 other, they describe the types of bonds. So even if this information which I presented you, they were not enough for you, you can find something additional in these films, or you can just repeat this material. And all these films, they are easy to understand, and well, I hope they will be helpful. Also, I found some page with tests, some kind of Quizlet. You can try to find something interesting in this page as well. So you see, if you want to enrich your knowledge, you should also find something by yourself. Me, I will prepare the questions for final test based on this information, which are in my presentations. But of course, you know, I'm not able to show you everything. So if you have some questions, you can ask, and I will try to answer them during the next lecture. And as you will see, there are still students who join this lecture. So at the same time, I should present you lecture. I should join the students, and that is why I'm not able to answer all these questions at the same time. Okay, so I hope all these topics for revision films, they will be useful for you. And last time I was talking about lipids, and I started to describe carbohydrates. So today I will try to finish this subject, and I will start the new group of biomolecules, which are called proteins. And last time I found such question. I told that carbohydrates, they are quite polar, and some person asked me what it means. So maybe quite polar. So it means these are compounds which are polar, but of course we can find other compounds which are more polar than carbohydrates. So I wrote that they are quite polar. And what it means that they are polar? It means they have polar bonds. And I was talking about such kind of bond. We should have some difference in electronegativity, the parameter which described each atom and its ability to form bonds. And in case of carbohydrates, we have, as you can see on this slide, we have many OH groups, hydroxide groups. They are functional groups, and they are polar groups. And that is why sugars carbohydrates, they are polar molecules, maybe not very, very polar, but quite polar. So it means polar enough to be soluble in water. And generally, such polar molecules, which have a special functional groups, polar groups, they are called hydrophilic compounds. It means that they are easily soluble in water. So I hope it's enough to explain why carbohydrates, they are polar molecules. Okay. Last time I was talking about chemistry of carbohydrates, so a special structure and special kinds of projections so that we can erode these structures using different methods. And I was talking as well about different functions of carbohydrates. So as you probably remember, in carbohydrates, they have plenty of such important functions. And today, few information about a classification of this group of biomolecules. The general, let's say, part unit of carbohydrates, this is one atom of carbon, one atom of hydrogen, and one OH group. This is a kind of unit, and of course we can find several such units in each molecule of carbohydrate. And as you can imagine, we can find simple carbohydrates, which are basic units to build more complicated, more complex structures. And that is why we can distinguish simple sugars, which are called mono and disaccharides, and the complex sugars, polysaccharides. What it means that in case of monosaccharides, they are built of basic units, means, for example, molecules of glucose or fructose. So these are single molecules of sugars, and they cannot be hydrolyzed into smaller units because they are the basic units. Generally, they are based on 3 to 6 carbon atoms. So it means that in their structure we can find 3, 4, 5, 6 carbon atoms. And depending on the number of these carbon atoms, we can talk about triose, tetrose, pentose, hexose. So depending on the number of carbon atoms, this is a special particular name of such groups of sugars. For example, in case of glucose, this is hexose. So it means glucose has 6 carbon atoms in its structure. This is in case of monosaccharides. Disaccharides, of course, as we can understand from the name, they are built of 2 such basic carbohydrates. And I will show you on the next slide what are the kinds of such disaccharides, very popular. Then we can find oligosaccharides. In this case, we have more than 2 basic units. So these are several covalently linked monosaccharides in the structure of oligosaccharides. So just it means that these are it means that such structures are more complex. And of course, as they are built of several smaller basic units, they can be hydrolyzed to smaller pieces. And polysaccharides, these are polymers which consist of different chains built of mono and disaccharides units, and depending on the structure, they are generally non-soluble in water, tasteless, and they can be hydrolyzed in some situations. So you can see, even if we have molecules built of the same basic units, they have, of course, different properties. So it means that depending on the structure, we can find different connections between such molecules, so different chemical bonds. Also, based on such special structures, we can find different properties. And as such molecules, they have different structures and different properties. They have also different kinds of functions. And here you can see the picture describing the formation of disaccharide, in this case maltose. And we have, once again, a kind of condensation reaction. So it means that in case of such kind of reaction, one molecule of water is produced and released. So as you can see, we have two single glucose molecules, and from the hydroxide group of one molecule of glucose, this hydroxide group is released, and from the other molecule, only hydrogen atom from this hydroxide group is released. They build a molecule of water. And after such process, these two molecules of glucose, they can join each other, and they are joined by a special kind of bond, which is called a glycosidic bond. So you can imagine that the formation of more complex structures in case of carbohydrates, it's like in the necklace, that we have single parts of the necklace, and they are joined each other, and they build some linear structure. Of course, not always this is a linear structure, but generally this is the simplest kind of complex structure of saccharides. So generally we have such linear polymers. And a few words about each type of saccharides, carbohydrates. This is the same. So first we have monosaccharides. And you can find here the structure of glucose and fructose. They have the same chemical formula, C6H12O6. It means they have 6 atoms of carbon and oxygen and 12 atoms of hydrogen in their structure, but as you can see, the structures are not the same. In case of glucose, we have some aldehyde group at the end of this molecule. Aldehyde group means COH. As you can see in the middle of this ellipse. And in case of fructose, we don't have such aldehyde group, but keto group. So here, the keto group is usually situated at the second carbon atom in the structure of this sugar, and such keto group, this is CO. So we have a carbon atom which is connected with two other carbon atoms and one atom of oxygen connected to this carbon atom. And in the case of such monosaccharides which have an aldehyde group, we can call them aldoses. And in case of such molecules with keto group, we can call them ketoses. And except glucose and fructose, to the group of monosaccharides belong as well galactose, mannose, and they are all hexoses. So it means they have 6 carbon atoms. So these are monosaccharides. So basically, which are then used to form more complex structures of sugars. But we can find as well some derivatives of monosaccharides. It means that the structure of monosaccharide is changed, transformed, very often is connected to other molecules, not from the group of carbohydrates. And what we can find here, for example, the sugars, which are the structural components of desoxyribonucleic acid, so DNA, and as we can find from this name, they have some additional groups, and I will describe you later this kind of sugars. Then we can find amino sugars when one or more hydroxylic groups, they are replaced by amino group. So we have one OH group is removed, and into this place amino group is joined. We can find also sugar acids, sugar alcohols. So in all these cases, one part of monosaccharide structure is replaced by another kind of functional group or another kind of chemical compound. On the next slide, you can find the structures of disaccharides. So such sugar structures which are built of two basic monosaccharides, 2 basic units, and they are important because they are really known. For example, in case of table sugar, which we use, the name of such sugar is sucrose, and this sucrose is built of one molecule of glucose and one molecule of fructose. Then we have maltose, which is called a malt sugar, and here we can find 2 molecules of glucose. And lactose, which is called milk sugar. Here we can find 1 molecule of glucose and 1 molecule of galactose. So in each case, we have 2 monosaccharide units, which are connected by a glycosidic bond. And in case of such disaccharides, which I mentioned, they are crystalline in form, they are water soluble, and they have a sweet taste. And you can find the structure of such disaccharides on the picture. So the main idea is the same, but just the basic units, they are different in case of all these three kinds of disaccharides. And very often in real life we can observe the process of souring of milk. It means that the milk sugar, lactose, is converted to lactic acid. So that is why the taste is changing from sweet to acidic. And we have completely different kinds of biomolecules in this case. And the more complex group are polysaccharides, which are also sometimes called glycans. They are, of course, polymers of monosaccharides, and such monosaccharides, they are connected by glycosidic bonds. And as such complex structures, they are really very big, and we can find also some additional bonds between such polysaccharides, so between the chains of these polysaccharides. These are mostly hydrogen bonds. So in consequence, we can observe different properties, physicochemical properties of such polysaccharides. They are not sweet. They are generally insoluble in water, and they are not in crystalline form. And we can find here two groups of such polysaccharides. One, they are called homoglycans. So in this case, they are made of only one type of monosaccharide monomers, mostly glucose, like we can find in case of cellulose, starch, or glycogen. So they are made of glucose units, but these glucose molecules, they are connected in different way. So I mean, they are connected by glycosidic bonds, but they are differently situated in the space, these particular glucose molecules. And that is why finally we obtain different structure. And the second group, heteroglycans, they are made of two or more types of monosaccharide monomers. And the example of such heteroglycans, these are histine, hyaluronic acid, or agar. And as I told before, depending on the kind of bonding between these basic units, polysaccharides have different properties. For example, as you maybe remember, I was talking about this last week. In case of starch, the bonds which are between the glucose molecules, they can be digested by our organism, human organism. But in case of cellulose, the bonds between glucose monomers, they are too strong to be digested by mammals, and that is why we are not able to digest cellulose. And here I know that this picture is maybe not very clear, but you can find different organization of such glucose molecules in case of starch, glycogen, and cellulose. So we can say that in some cases such polysaccharides chains, they are branched. So in case of starch, they are moderately branched. In case of glycogen, they are highly branched, and in case of cellulose, they are unbranched. So that is why such structure, and especially with hydrogen bonds between these particular chains of polysaccharides, this is a very strong structure. That is why it's not easy to digest such substance. And that is why cellulose is responsible for structural functions, and starch and glycogen not. They have different functions. And on the next slide, you can find once again a structure of starch and glycogen. So you can say they are the same, but the three-dimensional structure in case of starch and glycogen is different, even if they are both built of the same basic units. Okay, and I was talking during last lecture about cellulose, but once again, a few more information maybe. So cellulose we can find in plants only, and this substance is regarded as the most abundant organic substance in plant kingdom, and this is a main constituent of plant cell wall. That is why it should be strong, because it's responsible for structure of such plant cells. Cellulose is different from starch and glycogen because it's formed by beta glucose. So it means that the arrangement of these basic glucose molecules allows formation of strong hydrogen bonds. So this is what I mentioned before. And you can find here the picture of cellulose. So as you can observe, the neighboring glucose molecules, they have let's say opposite location. So they are joined up and down. You can see the groups CH2OH in every second molecule. They are situated on the top of this molecule, and between them we have glucose molecules with such groups which are on the bottom of this molecule, and that is why it appeared that such arrangement is really very strong. We can find also other polysaccharides like chitin, which is also very important organic substance, and is found in the exoskeletons of some insects, for example, or crustaceans. And chitin provides both strength of these exoskeletons and elasticity. And very often chitin is to heating, there are attached some additional substances like calcium carbonate, and in this case we can say that such structures of heating, they are impregnated with calcium carbonate. Okay. Okay, I can, yes, I can go back. Okay, here. Yes, they look similar, it's true, but the arrangement in the space of this particular chains of polysaccharides in case of starch and glycogen is different. And as you can see on the previous slide, in case of starch, we have a moderately branched structure. So you can see this is a little bit different than in case of glycogen, which is a highly branched structure. So just for us, maybe it's not a big difference because we can find more or less the same three-dimensional structure, but even such small differences, they are important. And you should remember that we have starch in plants and glycogen in animals. So they are, these two structures, they have similar functions, but one we can find in case of plants, another in case of glycogen. Okay. So I will continue. We have another kinds of polysaccharides. This which I mentioned before, so starch and glycogen. Starch we can find in plants, and is made of glucose units, exactly like in case of glycogen. But in case of starch, we can find some other structures which are called amylose and amylopectin, and they both are made of glucose. So in case of glycogen, we have just glucose units. So they are more or less the same, but first of all, they are found in different kinds of organisms, and maybe this three-dimensional structure is a little bit different. But they are both regarded as carbohydrate reserve. So they can be used as some source of energy, both for plants and for animals. And we have the last one, which is, I think, very interesting. And this polysaccharide is called inulin. So not insulin, which is a completely different structure, but inulin. And this is also a polymeric structure, but made of the molecules of fructose. So not glucose, like in case of starch and glycogen and cellulose, but made of fructose molecules. And such kind of polysaccharide, inulin, we can find in different vegetables like garlic, onion, leeks. You can find here the picture. And what is so special in this inulin? This is easily soluble in water structure, but not readily metabolized in our body. Also, it's filtered through the kidney. That is why one of the functions of inulin is just the usage for testing of kidney functions. But it's interesting that as inulin is not good metabolized, not well metabolized in our body, it means that it's not very caloric. So it means that we can eat a lot of garlic or onion, and even if we are on diet, and we are not able to observe some additional amounts of these substances stored in our body. So that is why they are really good if we are on diet. And also in case of garlic and onion, they have plenty of other very useful ingredients, very useful chemical compounds. That is why such kinds of food with inulin, they are very good for us, because they give us some additional important substances, but at the same time they don't build our body mass. Of course, they have special taste and smell, but who cares? We can eat a lot of such vegetables. Okay, so it was all which I wanted to present you about carbohydrates, so about saccharides. And now it's time to go to the next group of biomolecules, which are called proteins. Of course, everybody knows that there are such molecules called proteins, and everybody knows that they are really important for functioning of our body, and they have plenty of functions. And proteins, they are also a kind of polymeric structures, but as you will see later, this three-dimensional structure of protein is even more complicated than in case of fats and carbohydrates. And here you can find the picture, which I will show you once again later, and you can find different levels of structures in case of proteins. So we have primary structure, which then build a more complex structure called secondary structure. Then we can find tertiary and quaternary structure. So each next structure is more complicated. Of course, we know that proteins, they are maybe not the most abundant, but one of the most abundant organic molecules for us and for all living organisms. So not only for humans, but for animals, for plants as well. And in this case, the structure of the basic units which build the more complex structures of proteins contains not only the atoms of carbon, hydrogen, and oxygen, like it was before in case of fats and sugars, but we have additional kinds of atoms like nitrogen, sometimes sulfur as well. And it was found that proteins form about half, so 50% of the dry weight of the cell. And they are important because of many different functions, but they are important maybe first of all for architecture of cells, and as I told before, for many other functions. In case of proteins, they are also a kind of polymeric structures which are built of basic units, and in this case they are called amino acids. So I will show you later such structures of amino acids. And amino acids, they are also linked by a special kind of bond, which is called a peptide bond. And the main idea of such bond is the same like in case of sugars, for example. And once again, we can observe here, as you can see on the picture, the formation and releasing of one molecule of water. So this is a kind of condensation reaction once again. But of course, the groups, functional groups which can react to form such bond, they are different because we have an amino group NH2 and a carboxylic group COOH. But once again, from one group we can remove OH group, from another just hydrogen, and they both the species can form a molecule of water. So what are the special functions of proteins? Of course, they are responsible for nutrient storage, so they are necessary for different organisms to live. Then they can play very important function in the process of catalysis. It means that they can speed up some chemical reactions, which is also very important. They are responsible for functioning of our immunological system. They are responsible for transport of molecules, especially particular kinds of proteins which we can find in blood, and such blood proteins, they are very good transporters for other kinds of molecules. They play structural roles, so we can say they are structural support for all cells, and also they play the function in process of signaling. So they transport some electrical impulses between different parts of our body. So you see, in this case, in the case of proteins, maybe the functions are the most diversified in the case of this kind of biomolecules. And as I told before, we can find we can find basic units in structure of proteins, and such basic units, they are called amino acids. And in our body, we can find 20 different amino acids. And the amino acids, very often they are called the building blocks of our body as well. So we can compare amino acids to the words and to the letters, which can form some words. And the same is in the case of amino acids and proteins. And we have 20 such amino acids, but the main structure of each amino acid is the same. So as you can see at this picture, horrible. As you can find on this picture, in case of all kinds of amino acids, we have first of all the carbon atom, which is situated in the middle of such amino acid structure. And as carbon is able to form 4 bonds, we can observe this the same in this case. So it means that to carbon atom can be attached 4 different groups. And what we can find here, in case of each amino acid, we have amino group. As you can see on the left, so amino group, this is NH2. It means we have 1 atom of nitrogen and 2 atoms of hydrogen. The other group, which we can find in all amino acids, this is carboxylic group, COOH, which is built of 1 carbon atom, and to this atom they are attached 1 hydroxylic group OH and 1 oxygen. We can find also a hydrogen atom attached to this main carbon atom, and the last group is called the side chain, and in this case we have different such groups, so different for each kind of amino acid. And such amino acids, they are able to form more complex structures, like in the case of lipids and sugars. And such more complex structures, they are called peptides. And when such structures are bigger and bigger, we can talk about polypeptides. So it means that they are more complex, and polypeptides, they are also called proteins. So as you can see, amino acid, this is a building block, basic unit in case of proteins. And of course, we can find plenty of different kinds of proteins which are responsible for different functions. For example, we can find a peptide which is called aspartame, and this is a kind of sugar substitute. We can find as well enkephalins, which are responsible, which are a kind of pain relievers. So they are responsible for the treatment of pain. I mean, they can decrease the pain. And we can find as well such peptides like oxytocin, which is a kind of hormone, So it means it can regulate some functions in our body, and the oxytocin is especially important for pregnant women because it can induce labor in pregnant women, and it can reduce stimulate as well the release of milk during nursing. So as you see, plenty of different proteins, and all such proteins, they have different functions. And what was found that such function, this is the consequence of unique three-dimensional shape of a protein, each protein. So once again, we can find that, especially in case of proteins, that three-dimensional structure, three-dimensional shape is very, very important for different processes. So it means that sometimes we can find a similar parts in a protein molecule, but they form a different three-dimensional shape, and that is why we can find that such proteins, they are responsible for different functions. And here you can find such example. I will talk about this kind of process later during our lecture, so not today. And as you will see, we can find a special order of amino acids which are connected, which are attached to each other, and they create a polypeptide chain. And such order is called the sequence, and this is the primary structure. And such sequence can determine the shape of three-dimensional structure. So this, which is the most important for a functioning of each protein. And particular protein always has the same sequence of amino acids in the same organism. So this is such rule depends on the genetic information in our body. So in each case, when some particular kind of protein is produced, is synthesized in our body, they have always the same sequence of amino acids. And I don't know if it's clear enough, but we can find the process of catalysis here on this picture. So the violet structure, this is a protein, and the yellow shapes, these are some molecules. And as the protein is very huge molecule, we can distinguish different parts of this huge structure. And one of such parts can be a special binding site. Special binding site for a special kind of molecules. So it means that this is a site, location where we can observe that chemical reaction can occur. And so first, some target molecules, special molecules, they should find a right protein and the right part of this protein, which is called binding site. Then the structure of protein is changing a little bit. So it means that appropriate amino acids, they are positioned for this special reaction. So such parts of protein structure which are necessary for this reaction, they are positions, they are moved to the to these molecules which were joined to this binding site. Then we can observe the chemical reaction occurring in this binding site, and then the products of this reaction, they are released from this binding site, and the structure of protein once again is changed to this starting structure. So in this case, the protein, this is just such molecule which can help to occur the curing of some chemical reaction, but protein structure is not the it doesn't participate in a chemical reaction, but just it helps in this reaction. And such function and such proteins, they are called enzymes, and they can participate in the process of catalysis. So I will talk about this later, but I just wanted to show you that we have really different functions of proteins. Okay, but now I want to show you a few facts about the structure of amino acids. So these basic units which are the building blocks of proteins, more complex structures. Once again, you can find the basic structure of amino acids. So once again, we can find that amino group, carboxylic group, hydrogen, and some side chain. They are attached to carbon atom, and such carbon atom is called in different sources. You can find this is called alpha carbon atom. It means that to this atom they are connected 4 different kinds of groups. And depending on the structure and functions and properties of this additional side chain, we can find different groups of amino acids. So, of course, depending on the source, you can find different classification, but generally we can talk about 7 groups of amino acids. One is neutral, then acidic, basic, sulfur-containing, alcoholic, aromatic, and heterocyclic. As you can find here, the names of these groups of amino acids, they are based on some chemical properties of these side chains in case of each amino acid. And on the next slide, you can find this classification and the description of the nature of such amino acid, and as well the examples. So, in case of neutral amino acids, such amino acids, they have 1 amino and 1 carboxyl group, so 1 basic and 1 acidic group, and that is why they are neutral. And here we can find such amino acids like glycine, alanine, valine, leucine, isoleucine. And you can see as well some symbols of amino acids. So generally, when we talk about some amino acids, we can use the full names, but when we draw some structures, some sequences, we use only these symbols because it's easier, of course. You can find two kinds of such symbols. Before we used the symbols constructed using three letters, like you can see here, but the most modern approach is to use symbols constructed only from one letter. But I think easier to remember is such three letter symbols. I'm so sorry, you want to have break, but we will just finish, okay? Because then I have the next lecture. So just few more minutes and we will finish, okay? Because, you know, I have 3 lectures today, and just when I finish the one, after a few minutes, I start the other. Okay, so just 2 more slides and we will finish, okay? So you can find a description of this nature. So in case of neutral amino acid, they have one acidic, one basic group. In case of acidic amino acids, they have one extra carboxyl group. So it means that in the as some side chain, they have acidic group. And here we can find aspartic acid, asparagine, glutamic acid, glutamine. Then we have a basic amino acids. They contain one extra amino group in case of arginine and lysine. Then we have sulfur-containing amino acids, so they have such additional group with one atom of sulfur, and this is in case of cysteine and methionine. Then we have alcoholic amino acids, which have additional OH groups like serine, threonine, tyrosine. We have aromatic amino acids. They have cyclic structures, like in case of phenylalanine or tryptophan. And finally, a heterocyclic group of amino acids. They have additional nitrogen atoms in ring structure. And on the next slide, which will be the last for today, because I can understand you are very tired. So here you can find chemical structures of all these 20 amino acids. And as you can see here, they are, this is a little bit different classification. So they are divided into amino acids with non-polar side chains or polar side chains, or electrically charged side chains, which could be acidic or basic. But anyway, all these differences, they are the consequence of the fourth group in the structure of amino acids, so I mean the side chain. And you can find different colors of these side chain groups. So at home you can compare that really these are different kinds of additional groups, and That is why each of these amino acids have a little bit different properties, chemical properties. What is very important in case of formation of more complex structures? Well, okay. So I think that's all for today. So you have something to think about, and well, there are any questions?
Hello, ma'am. I want to ask,
Yes.
is it going to be objective or theory?
Theory. I mean, we are talking. I describe you only a theory, so we don't write some chemical reactions, or we don't calculate any examples. So it's just theory, but don't worry, I will explain you more precisely what you should prepare for this test, but maybe not next week, but before we will do this test, okay? Well, okay, so if you will have any particular questions, you can ask, you can prepare such questions, and for next lecture, or maybe not for next, for the next, and I will try to answer them. But I know that everything is very new for you and not so easy to understand, but don't worry. When I will discuss all these groups, you will see they are not so difficult. So just you should have some general meaning, general information, not so. Well, I show you some details because I think it's you don't need to remember everything. Okay, so thank you for today, and we will meet next week.
Dziękuję. Thank you, ma'am.